Good morning, everyone. I'm Tony Gover. I'm the Program Director for Bioengineering and Technology and our Core Injury and Repair program. So today I will be providing a brief overview of NEI's Translational Research Program, and specifically the R24 mechanism released by NEI. So there are many funding and resource opportunities for translational research within NEI and the NIH. Listed on this slide are some of the main opportunities for translational funding available to NEI researchers. So in addition to the translational research program I will talk about today, the NEI specific opportunities include our small business program that provides early stage capital for innovative small US companies, and our Audacious Goals initiative that is focused on regenerative medicine. Now, the AGI program does not have a specific focus on translational research. Its mission is actually much broader, but it does provide certain opportunities for translational funding. There are also a number of collaborative programs at the NIH that includes the Office of the Director, together with multiple NIH institutes and centers that support translational research. These would include the NIH Blueprint program that is focused on accelerating transformative discoveries in brain function in health, aging, and disease. And within the Blueprint program, there's the Blueprint MedTech program that is focused on translating medical devices. And then there's the Blueprint Neurotherapeutics program that is focused on translating biological and small molecular drug-based therapeutics. Now, one of the greatest advantages to working with these Blueprint programs is that in addition to the funding we can provide directly to the researchers, the funded researchers also have the opportunity to collaborate with NIH-funded consultants and can augment their project with NIH contract research organizations that specialize in things like regulatory policy, manufacturing, toxicology, and clinical testing. And this is at no extra cost to the researcher. So this program is particularly useful for products with, say, complicated manufacturing processes or a complicated regulatory pathway, or for investigators or groups that just have less experience with these types of activities. The BRAIN and HEAL initiatives are two other NIH-wide programs that, although not focused specifically on translational research, do provide certain translational opportunities in their areas of brain health and pain and opioid management. Finally, NEI works very closely with our partners at the National Institute of Biomedical Imaging and Bioengineering on encouraging bioengineering solutions for diagnostics and therapeutics by releasing multiple funding opportunity announcements. So you can find more information on all these translational opportunities on our website, but I would highly encourage anyone interested in these programs to discuss these opportunities with your program officer. These programs have very different missions and requirements, so you want to make sure your applications are aligned to the most appropriate program possible. So the NEI Translational Research Program is focused on translating innovative laboratory research findings into therapies for use by clinicians to treat visual systems diseases or disorders. And these therapies may be biologics, small molecular drugs, devices, or combination therapies. Now, we execute the Translational Research Program by releasing an R24 funding opportunity announcement, or FOA. And the focus of the application should be on obtaining the preclinical data necessary to support an IND or IDE application to the FDA. Now, we understand that there's an incredible amount of data that is required to submit an IND or IDE application, particularly for new regulated entities. And so the funding ceiling is very high for these awards. You may request up to $1.5 million per year in direct costs for up to 5 years. Additionally, since the application should be focused on translational research specifically, the scientific and technical merit review is conducted by a special scientific review group convened by the NEI for this very purpose. So we have actually had the R24 mechanism for many years, but prior to 2006, the R24 translational program was really focused more on creating infrastructure and core facilities for translational resources rather than a focus on a particular regulated product. So today, I will just be sharing information from 2006 to the present. And even from 2006, the R24 FOA has changed over the years, requiring more and more mature products as an entry point to the program, and a shift to products that have a clear medical indication rather than platform technologies. Okay, so since 2006, 20 awards have been made at an average cost of just under $9 million per award. Nearly half, 45% of these awards are still active. And so we should keep this in mind as we go through the next few data slides. Not surprisingly, the vast majority of awards are focused on biologics, with only 4 awards focused on small molecular drugs and only 1 device. And I will talk about that device in a few slides. Even though we only have 20 awards in this portfolio, the portfolio is very diverse. These awards are addressing over 13 different diseases, including diseases of both the anterior and posterior eye segments. So it is really great to see that our translational research program may have a great impact for so many diseases, and more importantly, on so many people's lives. Okay, our translational research program investigators have been very productive. So far, they have over 850 publications and at least 15 patents. And it should be noted that many of these investigators had secured their IP prior to their R24 award. So these patents are not included in that 15. And I will share one such project with you in a couple of slides. But this is actually a very good thing because secured IP, of course, significantly decreases the overall risk to product development. So far, our investigators have 8 granted INDs or IDEs for their original primary indications. And there are several products that now have additional approvals for other indications. And I will show you at least one example of this as well. There are now 7 active clinical trials, again, for the original primary indication. And we know of several more that will soon be initiated. And finally, we have 1 product associated with FDA market approval. And remember, 45% of our translational research awards are still active. So we expect these numbers to increase significantly over the next few years. So over the next 3 slides, I thought I would give you an example of 1 device, 1 small molecular drug, and 1 biologic that continues to progress towards the marketplace. So first, we have our single device. This original effort was led by Dr. Henry Edelhauser from Emory. And the focus of this R24 was to explore and develop novel transscleral drug delivery approaches to deliver drugs to the retina and other tissues at the back of the eye. In one approach, the team studied injection into the subchoroidal space, the space between the sclera and the choroid tissue layers, using a microneedle. The free length of these microneedles is only 900 microns, so the needles are very, very tiny. The suprachoroidal injection technique allows clinicians to target therapies to retinal tissues to potentially maximize efficacy while compartmentalizing therapies away from the unaffected vitreous and interior segments. This technique also allows clinicians to potentially deliver retinal therapeutics in an in-office-based setting. So to further develop and market this device, the PIs of this award started the company Clearside Biomedical. Their first FDA-approved product for suprachoroidal administration is Xipere. Xipere is a proprietary suspension of a corticosteroid, and it's indicated for the treatment of macular edema associated with uveitis. And as you can see from the slide, the company and their collaborators are pursuing several other drug combinations and indications with this microneedle device. This includes therapies for AMD, diabetic retinopathy, ocular oncology, and other retinal diseases. The second effort was focused on a small molecular approaches to restore vision for potentially numerous degenerative diseases. This effort was led by Dr. Russell Van Gelder from the University of Washington. The team focused on restoring light perception to patients with photoreceptor degeneration by making the retinal ganglion cells, or bipolar cells, photosensitive with the use of small molecular photoswitches. So photoswitches are pharmacological antagonists or channel blockers that use an azobenzene, a light isomerizable chemical moiety to block voltage potassium channels. So on the far left of this slide, we see an example of one of the photoswitches the team developed. In the presence of light, the compound is in the higher energy cis form. In this form, the compound can block potassium channels of the retinal ganglion cells or bipolar cells, depolarizing these cells and driving them to generate an action potential, making these cells light sensitive. In the dark, the photoswitch relaxes back to the trans form, allowing potassium to flow and hyperpolarizing the neurons. So the IP for this technology, which was secured prior to the R24 award, was recently licensed to a company for further clinical development. Now, Kiora Pharmaceuticals is planning clinical trials for this year with one of these molecular photoswitches. And it is for patients with late-stage retinitis pigmentosa. Okay, the third and last effort I will highlight is a biological therapeutic for achromatopsia. Achromatopsia is a rare autosomal recessive cone disorder with an estimated prevalence of 1 in 30,000. It typically manifests within the first few weeks or months of life, and affected individuals suffer from nystagmus, photophobia, severely reduced visual acuity, and color blindness. Now, 5 genes have been implicated in this disease, with a mutation in CNGB3 accounting for nearly 60% of those with the disease. And this gene encodes for the beta subunit of the cyclic nucleotide gated channel expressed in cone photoreceptors. So this effort was led by Dr. William Alsworth from the University of Florida. And this translational research award was focused on developing a recombinant adeno-associated virus gene delivery reagent for human CNGB3. So as a result of this award, the investigator was able to submit an IND application to the FDA, which was granted. And currently, Applied Genetic Technologies Corp., who was a collaborator in the original award, is conducting a multi-site phase 1-2 clinical study to evaluate the safety and efficacy of this genetic therapeutic. Okay, and so the takeaways. There are numerous translational opportunities available for NEI investigators. And we, the NEI, plans to continue to support translating therapeutics to the clinic and the marketplace. I would highly encourage applicants to reach out to us early in the process to discuss what opportunities and resources are available to help accelerate the development of potential therapeutics. And finally, please take advantage of these funding resources. I think one of the greatest threats to all of our translational programs is the lack of competitive applications. We actually receive very few applications for all of the programs I mentioned in the beginning of this talk. So the resources are out there, so please take advantage of them. And with that, thank you for listening, and I'm happy to take any questions.